Animal health. In agriculture, healthy animals are usually productive animals, so it's important that producers maintain their animals' health. Poor animal health can be detrimental to an operation when it causes a decrease in production levels and an increase in the cost of production. Producers constantly monitor animals for symptoms of ill health or any abnormal characteristics that an animal exhibits that indicates a bodily disorder. It's important to regularly use a health checklist to monitor vital signs, including temperature and respiration, and appearance of the ears, eyes, nose, mouth, and coat. Regularly scheduled visits from a veterinarian and good record keeping are also important to successfully monitoring animal health. Veterinarians are familiar with the symptoms of diseases common in different areas of the country. Disease is any variation from normal health that may affect an animal's anatomy or functions within its body. Diseases are directly caused by pathogens, which include bacteria, viruses, fungi, and protozoa. Parasites, such as flies, ticks, and worms, are vectors that transmit pathogens and often cause diseases in animals. If a disease is caused by a pathogen, then it is an infectious disease. A pathogen requires 4 things for survival. It must be able to gain access to the host body. It must be able to adapt to the host environment and multiply. It must be able to then leave the host, and it must be able to find another host to infect so that the cycle can continue. A disease transmitted from one animal to another in this way is said to be contagious. Some diseases caused by viruses are porcine reproductive and respiratory syndrome, or PRRS, which can cause abortion and weakened immune systems in swine, West Nile virus, which can cause encephalitis or inflammation of the brain in horses, avian influenza, which is transmitted from wild birds to domesticated birds and can cause loss in egg production and death, and foot and mouth disease, which can cause fever and blistery sores in cattle, swine, goats, and sheep that can result in losses in meat and milk production. On the right, we see the infrared image of a cow not infected by foot and mouth. And on the left, we see red hot spots on the cow that is infected by foot and mouth. Because these diseases are caused by viruses, they cannot be treated with antibiotics. Animals can be vaccinated against viruses, but often infected birds and animals have to be destroyed in order to eradicate the disease from the flock or herd. Foot rot, which is more prominent when cattle are kept in wet, muddy conditions, is very painful to the infected animal. Foot rot is caused by bacteria, so it can effectively be treated with antibiotics. Zoonotic diseases are those that can be passed between animals and humans. Some zoonotic diseases, such as rabies and Newcastle disease, are passed by direct contact between an animal handler and an infected animal. Other zoonotic diseases, such as West Nile virus, are passed between infected animals and humans by a vector that transmits the disease. Still, other zoonotic diseases, such as brucellosis, can be passed to humans who eat undercooked meat or unpasteurized milk from an animal infected by the disease. Environmental conditions called stress factors can stress an animal and predispose it to disease, especially if it is exposed over a long period of time or is exposed to multiple factors at one time. Extreme temperature, high winds, poor nutrition, mud, snow, dust, fatigue, weaning, poor sanitation, transportation, castration, dehorning, and abusive handling are all examples of stresses that can adversely affect an animal's immune system. Both stress factors and diseases can be minimized with preventative measures like proper nutrition and maintaining sanitary facilities. Nutrition needs of animals vary depending on the physical and environmental stresses they undergo. Pregnancy, lactation, and extreme temperatures all change an animal's nutrient needs. Undernourished animals generally have weaker immune systems that make them more susceptible to microorganisms. Sanitation programs are used to regularly clean and dry pen areas. Good sanitation prevents animals from being exposed to harmful microorganisms found in waste and in wet areas. It includes the use of antiseptics and disinfectants and the removal of rodents and dead animals. Animals should be purchased from producers that also have effective health management programs. If an animal's health does begin to decline, it will generally respond better to treatments in the early stages of sickness. Early detection of illness isn't easy, but some observable signs might be loss of appetite, listlessness, diarrhea, droopy ears, head held low, separation from herd or flock, coughing, and/or labored breathing. A sick animal's vital signs such as body temperature, respiration rate, and heart rate may be outside of the normal range. Losses in the animal industry can affect state and national economies. If the supply of a particular animal product decreases, then price for that product often increases. If one country has an outbreak of an animal disease, then other countries may ban products from that country in order to prevent the disease from entering their countries. And now, let's explore the core. Louis Pasteur was a 19th century French chemist and microbiologist who made advances in the fields of science, technology, and medicine. Pasteur is well known for discovering the process of pasteurization, where foods and beverages are heated to 120 to 140 degrees Fahrenheit in order to stop bacterial contamination. But he also developed vaccines for anthrax and rabies. As a scientist and researcher, Pasteur used the scientific method, the common steps that scientists use to gather information and answer questions. In fact, one of Pasteur's experiments helped settle debate over spontaneous generation, which is the belief that living organisms are created from non-living matter. Many people in Pasteur's time believed that cheese and bread, wrapped in rags and left in a corner, would spontaneously create mice because, after several weeks, one could find mice in the rags. Eventually, scientists were able to prove that mice didn't spontaneously generate, but some people still believe that microscopic organisms like bacteria did develop from non-living matter. Pasteur set out to disprove this belief. The first step of the scientific method is to ask a question or identify the problem. In Pasteur's case, the question was, can microorganisms really come from non-living matter? The second step is to form a hypothesis, which is an educated guess that can be tested. Pasteur's hypothesis was that living organisms didn't come from non-living matter. He also hypothesized that many bacteria were found on dust particles floating in the air. The third step of the scientific method is to design an experiment, and the fourth step is to conduct the experiment. Pasteur designed an experiment involving putting nutrient broth in 2 flasks. The necks of the flasks were bent into S shapes, and then the broths were boiled to sterilize them and kill any bacteria. The neck of one of the flasks was broken so that the broth would be exposed to the air. The neck of the other flask was left intact so that dust couldn't reach the broth. The flasks were allowed to sit for a period of time. The fifth step is to record the results and analyze the data. Over time, Pasteur noticed that the flask with the exposed neck contained broth that became cloudy. The cloudiness of the nutrient broth indicated that bacteria were growing in it. The broth in the flask with the unbroken neck continued to be clear. The sixth step of the scientific method is to draw a conclusion. Pasteur was able to conclude that microorganisms don't come from non-living matter, but instead come from other microorganisms. A sterile environment continues to be sterile unless it is contaminated by some outside object. Scientists don't stop after drawing a conclusion. They repeat the experiment many times to be certain the data is correct. Each repetition of an experiment is called a trial. Today's SAT word is antiseptic. An antiseptic is anything that destroys or restrains the growth of microorganisms that cause decay. In the lesson, we learned that an important part of raising healthy animals is the use of antiseptics that destroy or restrain the growth of decay-causing microorganisms.